I think that at this point, I basically have done everything that I can do in the browser. The next thing I will to demonstrate is how will you upload data to this data set, actually data files to this data set using the Riva upload tool. So imagine that you have done this, but you had to upload like hundreds of files. You are not going to do that with your browser one by one. Instead of that, what you want to do is use the upload tool. So in order to use the upload tool, the main piece of information that we need is to extract from this sort of data set what the structure is in terms of the identifiers for the data set and each of the replicates. Because like you said, files are always sort of associated to the replicate at this point. So we need to, in our machine, what we'll do is create in our hard drive, we'll create a folder structure that is organized by data set and replicates. And a lot of this information, very well described, is in our documentation. So I will go back. So I will do two things first. First, I'm gonna, since we haven't shown how to install these tools, I will take a little detour now and show you where the documentation is for that and how to install it in your machines. And then once I have it installed in my machine, I will come back to the data set, create the folder structure, and upload the data. So let's go back to the submit page, how to submit data, the phase-based curation documentation, and then we'll go to the section that says upload data files. In the upload data file, basically we already covered how to upload from the data browser. And the next section is organizing your files to use with the tool. And the way that files are organized in your local machine to use the Riva upload tool is basically creating a structure that has the top level directory has the read, what we call the read or the record identifier for the data set, okay? Which in this case was 1-RC7M, okay? That's the record identifier of the data set. And then for each replicate, you create a subfolder or a directory where the name of that directory is, again, the record ID or the read for that replicate. And then under that, you create a subdirectory called seq for sequencing files, a directory called proc for process data, and under that one, you will create another directory indicating what's a mapping assembly for that data has been processed with. So it can be MM10 or, HE38, this is human data or something else like that. Then you have, if you are uploading imaging data, you will create an imaging IMG folder where you will add the high resolution imaging files like the NIFTY files or the TIFF files, etc. Same thing for meshes. If you have, 3D models, you can add that under the mesh folder, and then you create a thumbnail directory where you will add the low resolution images or thumbnails of the higher resolution data. And the way that's organized is you have a thumbnail directory, and then under that one, a directory with the full name of the file, including the extension that thumbnail is derived from. And then in there, you put your low resolution images in either JPEG or PNG data. And same thing if you have microarray data or any other type of tabular data, you can put it under the array folder. Okay, so that's basically that very simple way of organizing the files. So the next thing, so this is, for example, how it will probably look in a given example. We're gonna do that now. So now let's move into installing the Riva clients. So the main information about the Riva clients is also given in here. We have distributions of these client tools for Windows and Mac. We have Windows and Mac releases, or you can install it also on Linux using the pip facility. So you have, if you have Python 3 installed, you had to have Python 3 installed in your machine, and then using pip, you can install all these clients. And I will mostly demonstrate the GUI version of the tool, but I want to point out that there's also a command line version of these tools. The documentation shows exactly how to use them with the command line tool if you want to upload the data. Let's say you have all this massive amount of data in a cluster and that runs Linux, and you don't want to use a GUI, and you don't have a GUI in that cluster, then you can just do command line, upload all these files. So I will demonstrate it I will install the tools in my Mac. So I go to the Riva client release page. I'll download the version for my Mac. I start the installer. I typically get this notification. It's not from a trust developer, so I need to go back into my system preferences, go to security. And just follow the installation defaults. And in my machine, this gets installed under applications. You will see the Riva client tool folder, and under that folder you have all the different tools. So the DB back tool is the one we demonstrated earlier today, but there's also the authentication agent that can also be used, for example, especially if you are doing the command line version of these tools, you will probably have to use first the authentication agent to generate a token, and then once you have that token, you use the command line interface to upload the files. But now what I will demonstrate is the Riva upload tool. So I will start the Riva upload tool. But it starts. So now I will pause the tool for a second and move back into the data set and into creating in my machine the directory structure that I will use in order to upload the data. So if you remember that picture from the documentation, we need to create this directory structure where the top level folder is the record ID of the data set, and then one directory for each of the replicates. So I sort of have already created a sort of a folder structure here that has that is structured in the way that I want the data to be. The only thing I need to do is replace or substitute the name of the reads. And this is actually this directory structure and data is the one that we distribute for the self-guided tutorial. So if you want to, at the end, want to do the self-guided tutorial, you basically will have to go through this exercise. Do you download that file, expand the zip file, and then you just do the same read substitutions that I'm going to do now. So the first thing I need to do is change the name of this, the top folder, enter the read of my data set. So I go back to my browser. I can copy the record ID of that my data set, and I enter it in there. The next thing I want to do, I will need to get the replicate for the, let's say, the micro CT control. So the replicate associated to the micro CT control is this one, 2-HW1J. So I will copy that one, and I will name the directory for the micro CT control with that, to that number, to the name. I will do the same for the micro CT mutant. Oh. Oh. I guess that. Okay. Try it again. And then I will do the same for the RNA-seq replicate. And now I have the data organized under this directory 2-HWP-W0P in the way that I want. In a real case, you will not do it this way. You know, in a real case, you will start from the top. You will, okay, first go to your data set, copy the read of the data set and create one directory with that name. Then you will start creating one directory for each of the replicates. And then under each of these replicate directories, you will start creating either the thumbnail folder or the imaging folder or the sequencing data and the sequencing data folder and the process data folder. And then you will start moving your files to that location. So I already have that structure built in. So I have under each of the replicates, for example, if I go to this replicate, I have one folder for my images. So these are going to be the high resolution images. And then I have a folder called thumb for thumbnails. And then under the thumbnail, I have another folder with the actual name. The name of the folder is the actual full name of the file that this image is a thumbnail of. And then I have the actual low resolution image. Same thing for the, if I go to this replicate, I have one folder for the process data. Under that folder, I have a folder for the assembly that was used, in this case, MM10. And then under there, I have all the files, the processed files. I mean, we have the TSV files, some bigwig files, and some BAM files. And then under the sequence, under the seq folder, I have the FASTQ files. So now I basically have the data, I hope, in the way that it's supposed to be. So now I will go back to my tool and try to upload the data to the data set. So when you start the tool, the first thing you will do is check in the options that the tool is actually pointing to the correct server, basically to www.facebook.org. The next thing you do is you log in using the same identity that you use in order to create the data set. And then the last step, once you have logged into the correct server, is click the Browse button, navigate to the top level directory that has the record ID of the data set, which in my case, it was this one. Click open. So now the tool will scan that folder and see what are all the files that it finds under that there. And it does a little bit of a verification, and then it tells that it's ready. And then you just click the upload, and it will start going one by one, uploading the files. And as you can see, it gives you a very detailed explanation in here in the console of what happened. If you get any errors, you will see the errors listed here in the console, so you can communicate to us. Like if you're having problem uploading the files, then you can copy the error messages here that are given in the console and email that to us, and we can see what's going on. Another very important, one of the most important features of this tool is that, it will continue uploading from where it left off. So, we live in this sort of remote world where you're always uploading data and large number of files and large files from remote locations. You can lose your connection, and then you don't need to restart the whole process. So in case you get disconnected, then you go back, you rescan your directory, you restart the upload, and it will continue uploading from where it left off. So, so that's a really very, very important feature. So now if we go back to our data set, we'll see that all the thumbnails and the images, the imaging data, the additional sequencing data, the process data, and the track data are there. So imagine that you have hundreds of thousands of files. This can be very, very useful. So I think at this point, I'm pretty much done. The last thing that I would probably do in this type of experiment is, since we are uploading track data, bigwig files, then one thing you can do if you want to see this data automatically display in the SC, the Santa Cruz genome browser, you can add a genome browser entry here, indicate what was the mapping assembly, or if you have more information about the chromosome and start position, end position, you can also enter in there. If not, just the mapping assembly, and you create that entry, and then the data set will feature, probably after a nightly run, it will show the tracks, your bigwig tracks in the genome browser. So you don't need to have your own track hub or anything like that. You can just directly upload your track files in here and directly visualize them and see them in the Santa Cruz genome browser. So I think that that's pretty much everything I wanted to demonstrate. I don't know if you have any questions about the whole process. So maybe I will end by probably going back to the project, showing that I had created this Bootcamp self-guided tutorial data set that is pretty much the same. So you can follow all this information. And if you go down here to the supplementary files, it has a zip file with all the data and then some instructions on how to do the tutorial. So basically we will do exactly the same thing I did. You will join the demo group, then you will create a new data set, add all this information, download the zip file, expand it, change the name of the directories to match the record IDs of the new data set that you created, and then use the upload tool to upload the files. So, great. Any other question? Has there been any questions on the chat? I haven't been monitoring.
No, nothing new since the last one. Is anybody planning on submitting data to FaceSpace in the near future? I recognize Michael Payne as one of our guests here, and he's already part of the process. So we're glad that you could be here. And yeah, if you have any questions, if you try the self-guided tutorial yourself and have come across any issues or confusion, definitely please feel free to contact us. The best email for that I'm going to put in the chat, which is help@facebase.org. And we're here to answer your questions. So if there are no further questions, then yes.
Chris, I just want to say thank you. I've got a few people from my group here, and we will be starting to upload data in the next couple of weeks, I imagine, with Fu at USC. So I think we'll be okay, but I really appreciate you and your team spending this time talking to us today. It's, we've got on, I have an account, but we haven't really uploaded any raw data yet. So that will be something we'll start doing in the next few weeks, as I say.
Okay, great. So hopefully it's a little clearer seeing.
Yeah. And also with with many of the other groups, it's very useful. Like we have a short session and we discuss exactly what are the files that you're going to upload and more or less what the structure of your data set is, and we kind of guide you through that process and to make sure that you start going in the right direction. We found that very useful, and they've been able to upload a large number of data and files pretty rapidly.
Well, thanks very much. I appreciate it.
Just send us email whenever you're ready.
Yeah, that's what